All right, thanks guys. Thanks for joining us here this morning. Apologies for getting the meeting started a few minutes late. I think we had some scheduling issues this morning, but we'll go ahead and get started here momentarily. I'm gonna go ahead and pass things off to Marta and have her make an introduction to Dr. Clement for us this morning. And so with that, Marta, are you able to speak up?
Can you hear me, Peter?
Yes, I can.
Okay, then we are excited to continue the SRF webinar series. The goals of the series are getting you closer to the science, making you aware of the research that is being done and the opportunities to participate, and empowering your communications with clinicians. We also want to remind you of our next webinar in the series with Dr. Heather Medford on Thursday, October 15 at 10 a.m. Pacific time, titled SYNGAP1, the road from gene discovery to targeted therapy. Today's talk is towards therapeutics for autism spectrum disorder using SYNGAP1 heterozygous mouse model by Dr. James Clement. Dr. Clement has had a long career in research and particularly in neuroscience. First, in the MRC Centre for Synaptic Plasticity in the UK. Then he took a postdoctoral fellowship position with Dr. Gavin Rumbaugh at the Scripps Research Institute in Florida from 2010 to 2013. In the Rumbaugh lab, Dr. Clement dedicated his time to study SynGAP and was first author for a groundbreaking paper with the publication done in 2012, Pathogenic SYNGAP1 Mutations Impair Cognitive Development by Disrupting Maturation of Dendritic Spine Synapses. And SYNGAP1 links the maturation rate of excitatory synapses to the duration of critical period synaptic plasticity in 2013. He also participated in one of the most comprehensive reviews about SynGAP, which many families have read, Syngap1 Mind the Gap, published in 2016. He then went back to India and got his own lab at the Neuroscience Unit, Jawaharlal Nehru Center for Advanced Scientific Research. His lab is dedicated to neuroscience, especially in autism and intellectual disability using Syngap1 model. His interest is to see how SynGAP deficiency impacts synaptic structure, function, and maturation, particularly during early stage of development. Needless to say, Dr. Clement must be one of the most knowledgeable people in SynGAP. As a group, we are immensely thankful about Dr. Clement's contributions, collaborations, and expertise in SynGAP. We truly enjoy working with him. After this brief introduction, I want to let you know a recorded version of the webinar will be available on the SRF website. By the end of this presentation, you will have opportunity to get your questions answered. We love to hear from you. Please write your questions in the chat. For those of you just joined us, welcome to our talk today towards therapeutics for autism spectrum disorder using SYNGAP1 heterozygous mouse model by Dr. James Clement. Dr. Clement, go ahead, please. Thank you.
Thank you very much, Peter, and thank you, Martha, for the introduction. Let me just share my screen. Right, it's a pleasure to meet the members of the SynGAP Research Foundation. I thank Mike, Peter, and Martha for this opportunity to discuss one of the major findings from my lab. So briefly, my lab works on SYNGAP1. So we try to understand how environment and genetics interact with each other to trigger intellectual disability or autism spectrum disorder. So to understand intellectual disability or autism spectrum disorders, we should try to understand and study how the neurons and non-neuronal cells are formed during developmental stages. So in today's presentation, I'm going to discuss one of my graduate students' work, Vijaya Verma. It was completely done by her. and some of the experiments were assisted by one of the other graduate students, Mr. Vijay Kumar. So, like I mentioned earlier, to understand intellectual disability and autism spectrum disorder, we need to study how the neurons are formed during developmental stages. If you look at the development human neurophysiological aspects, an adult human brain has roughly 86 billion neurons, and there are around 885 billion non-neuronal cells. It is important to understand and study their function and how they communicate with each other, mainly from birth till the end of the adolescent stage. It doesn't look that easy. The reason is, if you look at a newborn baby's brain, it might have the number of neurons that you might see in the adults, but the connections are very less, and many of the neurons are inactive. But by the time they reach 3 years of age, there's a huge increase in the number of connections, but those connections are not completely strong. But by the time they reach the adolescent age, the connections that are not activated repeatedly are lost, and the ones that are repeatedly activated, they are strengthened, and we can remember those things for a very long time. So one of my favorite example is, even though we are about 25 years or so, we still remember what is a plus b the whole square stands for, and what is the result for a plus b the whole square, even though I haven't done real mathematics for many decades. So to understand how this formation and elimination occurs during development, whether these formation and elimination are happening at the same time in different regions of the brain, or whether they happen during different time points, starting from birth till the end of the adolescent, and maybe during to a certain level in the adult stages. This diagram illustrates the pattern of maturation that happens in the different regions of the brain. So the first region, the region of the brain that really develops properly, is the senses: vision, hearing, and touch. And by the time we reach the childhood, we begin to talk properly, and we're able to construct a sentence correctly. And you can see here at birth, the number of neurons are very less, but there is a window when it peaks. That means the neurons at this region is extremely active. They were there. If when you look at the babies, by the time they're 1 year or 1.5 years, they're very, very active. They're very responsive to sound. They're very responsive to touch. They look around everywhere. So that window is very, very critical. And at the middle of childhood, or a little bit later, the region corresponding to the senses pretty much closes. That means the opportunity to rewire or change the connections based on the external stimuli is very, very negligible and minimum. It's very difficult after the time point. But the interesting aspect is, if you look at the adolescent time point, end of adolescent, that is roughly around 18 to 22 years, the higher cognition and the language does not completely come to zero. So that tells us that even though the window is pretty much past, we are somewhere here, we are still able to learn and remember things, even though we cross the peak point of those connections, neuronal activity and the connections. So for example, if you make a mistake in an adult stage, we are still able to modify our mistakes and then correct our mistakes and then fix the problems. And when we migrate to another place, we are still able to learn the new language. So this whole process of formation, elimination, and maintenance of connections are mediated by hundreds of proteins present in the pre-synapses as well as in the post-synapses. So the purple ones are the ones that are already implicated in autism spectrum disorder, and one of the major ones is SynGAP1. So today we're going to discuss about the SynGAP-associated intellectual disability and autism spectrum disorder. So if there is a mutation in a gene encoding any one of these proteins, it can disrupt the normal function of synapses, and that can result how this window is operated. This window can be shifted to the left, or this window can be shifted to the right, and that will disrupt how the brain develops, and that can lead to long-lasting consequences, which we see in many of the autistic children with autism. So how this window period might affect the brain development? As I mentioned earlier, this is the normal pattern of brain development from birth till 15 years of age. But in intellectual disability or autism spectrum disorder, it can lead to hyperconnections or hypoconnections. So why it matters? People might ask, okay, if you have hyperconnections, that means you should be able to respond better compared to the normal healthy person, or in the hyperconnection, you may not be able to respond properly. So what happens is, in the normal brain development, the information is processed accurately. Whereas in cases of intellectual disability or autism spectrum disorder, the information is not processed properly. So the output by the kid or the adult person is not correct. They're not able to sense how to respond correctly or able to learn things and respond appropriately. So the major challenge facing scientists who are working on autism is to restore these abnormal connections or information processing and make those kids or the patients of autism to able to process those information. We need to correct this lack of information so that they will be able to understand and follow all the norms of society, and pretty much they'll be able to do everyday activities. That has been a challenge for many decades in the field of autism. So why we need to study autism? So to understand whether we can correct the phenotype, particularly after, like, let's say 3 to 5 years of age, by the time majority of the brain regions are pretty much developed, we need to understand how neuronal and non-neuronal system functions and develops during from birth till 18 years of age. So for that, we use Syngap1 as a model. Like Martha has introduced, I have shown what happens to the brain development during SynGAP mutation, and follow-up studies from Gavin's lab and Richard Hugon's lab have extensively hypothesized and proved how SynGAP mutation disrupts the network activity, development of the neuronal cells, but not much has been known in terms of the non-neuronal cells. So, it is evident from the previous studies, starting from Hamden's work, that SynGAP haploinsufficiency or nonsense mutation or frameshift mutation leads to various behavioral disorders, predominantly intellectual disability and autism spectrum disorders. However, the current therapeutics are aimed at alleviating some of the major problems like sleeping issues or epilepsy or eating problems, but the current drugs are not really aimed to correct intellectual disability or autism spectrum disorder and other related behavior issues. So we understand from my earlier study that one of the reasons to see such behavior abnormalities in patients of autism or intellectual disability is due to altered synaptic and neural network function. For example, when we activate one specific neuron here using uncaging of glutamate, so this is a hippocampus from an acute brain slice hippocampus from a wild type as well as from a mutant mice. And this is completely soaked in the voltage dye. And we have done, this was done in Gavin's lab, and we have done a 2-photon imaging. So when we activate one specific neuron, under normal conditions, that activity might spread to a particular or a certain region of the brain. It will not spread to many other irrelevant regions. But that regulation is completely abolished in the heterozygous mutation. So you can see in this video that activation of one specific neuron leads to spread of activity within a certain region. But in the mutant, you can see there's a huge increase in the spread of activity. So that regulation to control the spread of activity is completely lost in the heterozygous mutation. That one reasons why they're not able to process information accurately, why they have other behavior issues, why they, this might also explain why they have epileptic seizures and delayed language problem, because when you have a spread of activity like we see in the heterozygous mutation, that may lead to abnormal processing of information. So based on this particular study, we hypothesize that this dysregulated or lack of control over the spread of neuronal activity might be due to the disrupted GABAergic function. So people understand that GABA, when you talk about GABA in adult stages, everybody thinks that it is the inhibitory neuron. However, GABA has dual role. At the early stages of development, it has to play a role of activating the neurons, not suppressing the neurons. Whereas by the time the brain matures slowly, it needs to convert it from activation to suppression of neurons. So this kind of polarity switch from activation to inhibition is mediated by a couple of chloride cotransporters. So these chloride cotransporters are called NKCC1 and KCC2. So at birth, there is an increased level of expression in NKCC1, and that expression goes down as the brain matures, and it is replaced by KCC2. So KCC2 has to form oligomers in order to function. If it fails to form oligomers, then there'll be abnormal level of chloride concentration inside the neuron, which will be detrimental for the neuronal development. And if the switchover between NKCC1 and KCC2 does not happen, that can also lead to various developmental disorders. One of the major ones is neonatal epilepsy. So the function of this NKCC1 and KCC2 is to maintain the chloride concentration inside the neuron when there is no activity, that is at rest. Here you can see at the early stages of development, there is NKCC1 allows more chloride to enter the cell, so the chloride concentration is very high. When the chloride concentration is very high, upon activation of GABAergic neurons, when GABA binds to those receptors, it leads to chloride. It allows chloride to leave the cell, and that depolarizes the neuron. So that means it activates the neuron positively. While that happens at the early stages of development, when the brain is exposed to the external stimuli more frequently and everything is going fine, this switchover from NKCC1 to KCC2 happens. And the function of KCC2, once they form oligomers, is to remove chloride from inside the neuron, thereby maintaining lower concentration of chloride ions inside the neurons. And that level is what you would find in an adult brain. And when we have lower concentration of chloride inside the neuron, when GABAergic neurons are activated and GABA binds to the respective receptors, it allows chloride can enter the cell and thereby suppressing the activation of these excitatory neurons. So it plays a dual role. At the early stages, it activates the neuron, whereas at the later stages of development, it suppresses the activation of neurons. That means it's inhibiting the neurons. So why it is very important to maintain the switchover? As I mentioned earlier, one of the major issues with neonatal epilepsy is the abnormal switch over between NKCC1 and KCC2. And this is a typical responses that you would see in a normal brain. And when that switchover does not happen, it can lead to high intracellular chloride level, or if you have more KCC2, it can lead to low intracellular level. And that can disrupt how the GABA functions in the neuron or in the brain. So it can either excite or suppress too much, and that can lead to extremely abnormal synaptic function. And that will disrupt how a person processes an information when a particular stimuli is given. Apart from that, it can lead to altered excitation and inhibition. Here you might have more excitation compared to normal conditions. So it can either make the brain more excitable or more inhibitory, depending on whether the switchover happens at the right time, or it might happen earlier, or it might happen little later. So several studies from other models like Rett syndrome and Fragile X syndrome have looked at these GABA polarity carefully, and they found that from the human patients, this expression of chloride cotransporters is reduced in the Rett syndrome, and this has been replicated in the mouse model also. In the Fragile X mouse model, so this is in the somatosensory cortex, the P stands for the number of days after birth, and the maturation time point for somatosensory cortex or the barrel cortex in the mouse is approximately 7 to 9 days. People can look up to like 11 days. So that's the reason you see up to 15 days here. Whereas in the hippocampus, when we discuss my data, it will be a little bit longer. So I'll explain that when we come to those slides. And they don't see a change in the KCC2 expression, but you can see here towards the end of the maturation time point, there is a huge increase in the level of NKCC1, and that has resulted in the abnormal synaptic activity. So minus 60 is the resting membrane potential. That means the membrane potential to be maintained when the neuron is not activated by any stimulus. And you can see in the black one, the wild type, it reaches the adult level by 8 days after birth in these mice. But in the Fragile X mutant, it is delayed by at least a week. It reaches the adult stage by 14 days. Because of this shift towards the right side, all the synaptic connections might be disrupted and dysregulated, and the expression of certain receptors might also be affected. So not a lot of complex activities will be affected because of the switchover, because of the failure of the NKCC1 to switch over to KCC2 expression. Right. So from these studies, many people have tried drugs that might be targeting the GABA receptors. Here in this example, there are many drugs that are available, like diazepam, which is a well-known drug, and bumetanide is another well-known drug that can regulate the GABAergic function. And in this case, bumetanide is targeting the NKCC1, and by blocking it during this developmental stages, it can somehow restore the synaptic function to the normal level. So based on this particular hypothesis, they went back to the Fragile X model, and what they have done is in the somatosensory cortex, like I mentioned, the developmental time point is roughly around 10. If you remember my previous slide, we see abnormal expression of NKCC1 in 10-day-old mice. So what they have done is they have given bumetanide from birth till 10-day-old mice, and they measure the membrane potential. They measure the chloride concentration. So this is E-GABA. So the resting membrane potential will correlate to the amount of chloride present inside the neuron. More positive means more chloride. If it is around minus 60 means it is less chloride present inside the neuron. Here you can see in the FMR1 normal mice, bumetanide doesn't make any difference. Whereas in the knockout, it is more positive. But when the mice that received the bumetanide treatment, they were able to restore the function back to, they were able to restore the membrane potential back to the normal level. That by it might help in correcting the phenotypes associated with Fragile X. However, in this paper, they haven't really looked at the whether application of bumetanide to these mice restores or corrects the phenotype deficits observed in Fragile X model. However, I just want to mention that some of the drugs that I'm going to talk about does not target NKCC1 or the NKCC1. So the major question, like I mentioned in the beginning of the talk, is what happens when we give the drug after this time point? Because by the time you take a child to the doctor, like I mentioned in the introduction, some of the brain regions are already developed. Then how do we restore or correct those phenotypes? That's the biggest challenge. Most of the studies that have tested many drugs, they target only the developmental time point. They don't really test what is happening after the developmental time point. Can we correct them? Can we make the mice or the patients lead a normal life every day, at least to 70 to 80%? So that study is not very conclusive from many different mouse models as well as from many different drugs. So that's one of the reasons I believe that the drugs fail in the clinical trials, because we take these drugs based on the study performed mainly during the critical period of development. We ignore the most important part is what is happening after the critical period. When we give the drug after this time point, do we see this kind of result or is it not effective? So that's one of the biggest challenges facing researchers in autism. To understand whether we can also try that in the SynGAP mice, we went ahead and checked what is happening to the GABA polarity during development in SynGAP mice. We did the same experiments where we looked at the chloride levels inside the neuron, like I showed you earlier. This is the normal resting membrane potential of resting membrane potential, whereas when we have the chloride co-transporter mutants, you can see that the resting membrane potential is shifted towards more positive, which is much closer towards the firing threshold. That means you need a little bit of stimuli to generate activity in the neuron, which is not the case in the normal brain or a healthy brain. So the challenge is to see, can we restore the more depolarized or the ones which are closer to the threshold back to its normal level, which is between -60 to -70. So if you see any abnormal level of resting membrane potential, like here in this case -55, that suggests there is an excess level of chloride concentration inside the neuron. So when we did those experiments from different age groups, we observed that the blue one is for wild type and the red one is for the heterozygote for the SynGAP mutant mice. Here you can see in 2 weeks old mice. So these are all done from the hippocampus region. So these are acute brain slices, so that we can keep them alive for like 10 to 12 hours. We have done this is done using a gamicidin patch clamp technique, so that we don't alter the level of chloride concentration inside the neuron. We preserve the actual concentration which is present inside the neuron, so that it will be a more direct correlation to the function of GABA, whether it is excitatory or depolarizing. So in this case, you can see in 2 weeks old mice, the neurons present in the SynGAP, they have more level of chloride, which is correlated with more depolarized resting membrane potential that is more positive here compared to the white, which is more around -65 to -70. And so, we, from my studies in Gavin's lab and Gavin's recent paper on the human iPSC lines, we know that these the SynGAP gene has a maximum effect in the 2 weeks old hippocampal region. And that is correlated here very, very well. So it could be because of we see an increased activity. There is some kind of compensation might be happening with the GABAergic function, and that is going completely wrongly. That might lead to altered switchover between NKCC1 and KCC2. So this result gave us an idea that the GABA polarity switch might be completely affected in SynGAP mutation. So based on the earlier studies and the hypothesis that GABA might be a good target, so we had a drug, a compound, I should say, not a drug, compound in our neighboring lab, which we know has a potential to correct the synaptic activity in the brain of in the SynGAP mutant, as well as in the other models of neurodevelopmental disorders like Fragile X and the Rett syndrome. So the question we asked is, if we inject this particular compound, where can we restore the GABA polarity more to the healthy brain? If that is the case, that'll be amazing. But the biggest challenge comes is whether it can correct the phenotypes in these mutant mice. So we did 3 different strategies. One of them is by injecting only during the developmental time point. Like I mentioned earlier, we use hippocampus. In the hippocampus, from my earlier studies and other studies, we know SynGAP unleashes a detrimental effect between 14 to 16 days after birth in mice. So we want to target that region, and we also, in the KCC2 or the chloride co-transporter expression, we do see abnormal resting membrane potential at this specific age group. So the first strategy, like people have done it for Fragile X, is to target only the developmental time point, and then see when we administer the compound only during the developmental time point, is that enough to correct the phenotype even when the animal reaches the other stages. So this group is pretty much corresponding. It's an approximate age group, equivalent age group in humans. And we evaluated the effect of the drug in correcting the phenotype using open field social isolation and novel object recognition. I'll briefly touch upon the rationale behind doing these behavior paradigms. So before we were tested for behavior, we asked the question whether injecting the drug is during this particular period is sufficient to correct correct the synaptic function, that is to restore the membrane potential back to the normal level. So like I showed you earlier, this data further confirms or validates our finding that in the heterozygous mutant, we do see more depolarized neuron. That means more chloride is present inside. And when we administered the compound, it does nothing to the wild type, which is a really good sign. There is no side effects or any secondary effects of the compound. And in the SynGAP mutant, it is correcting the synaptic function by restoring the resting membrane potential back to the normal level. You can see that this is more depolarized. This is more depolarized, and this is more closer to the resting membrane potential like the wild type. So we don't see any significant difference between these two groups, suggesting that there is a complete rescue in the SynGAP mutant mice that received the compound. So this was very encouraging. But the most important question is, neural functions are corrected. That is amazing. But from the literature, we all know that when you see a change in the synaptic function, it's not necessarily should reflect on the phenotype, like the behavior, restoration of behavior. So we went ahead and asked the question, what happens to the phenotype of these SynGAP mice that received the drug? So we did 3 different behavior tests: open field test, social isolation test, and the novel object recognition test. So open field test talks about the locomotor activity. We know from the many studies, patient studies, that majority of them are hyperactive and they're less anxious. So these mice tend to run closer to the wall because they don't want to go to an open space in fear of being picked up by the predators. If you see rats running on the road, they will run on the dark regions and more closer to the wall because they know if they go to the center, they have a chance of being picked up by the predator. And they don't go to the center also much because like I said, they don't want to be picked up by the predator. So if they go to center of this open field mode, that will correspond to the anxiety level. So if they go to the center more, that means they're less anxious. Whereas the normal mice, they don't really enter the center that much. For example, in this case, if they do 20 times, the normal mice might do only 2 or 3 times. So that will indicate, so any change when we give the drug would results in a less number of entry or running around corresponds to the normal activity. We will know that the drug has an effect. And we did the novel object recognition. It's a memory test. It's a very simple memory test. We trained the mice for 2 familiar objects. And after 24 hours or a few hours later on the same day, when you replace the familiar object with a new object, the mice will always should be able to distinguish between the familiar object and the new object. So this we will know by the time it spends with the new objects, because they are very curious. Like humans, these mice and the rodents are very curious, and they want to see what is the new object which is lying there. And the final behavior experiment that we did is the social normality test. And we know from the studies that the patients with SynGAP mutation or Fragile X or Rett syndrome or any other mutation implicated in autism, they struggle to communicate with people, or struggle to make friends, or even with their cousins, they struggle to talk to them. So this experiment kind of somewhat recapitulates that scenario. First thing, and also the patients with autism or intellectual disability, they tend to play more with the objects and avoid contact with their people. So this experiment paradigm, behavior paradigm, somewhat recapitulates the same situation. So what we do is we train the mice in this three-chamber. We train the mice, make them explore it. Once it is explored for a few minutes, we introduce an object and a mouse there. Usually what normal mice will do is they will spend more time with the mouse, not with the object. And when we introduce a new mouse, after a few minutes, they will tend to spend more time with the new mice. So this is more like what we do in the lab or what we do with friends. So when we meet new friends, we try to, and we are curious to know who they are, where they are from, what they do, why do they come to this city, all the questions you want to know about them. So the mice also, similarly, the mice will also try to explore what is this new mouse, who is he or she, what are they doing here. So they'll spend more time with the new mice. Whereas in the autistic mice, mice with SynGAP mutation or models of autism, they will tend to avoid, sometimes they avoid both the mice or try not to spend more time with the new mice. So again, the time spent with either this mice, old mice, or the new mice, or the object or the mouse determines what is their social activity and how it is processed in the brain. When we did the open field test, we were able to recapitulate all the published results. You can see here in the wild type. So to make things easier for you, blue color, light blue, it always corresponds to wild type. Red one always corresponds to SynGAP. And I have categorized these two groups as vehicle. And hopefully then when the animation comes next, you will be able to see the results for the compound. And you can see here, these SynGAP mutant mice, they tend to be hyperactive. They travel more distance in a given time compared to the wild type. And when we give the drug does not have any effect on the normal mice. But the good thing is the drug is able to restore or correct the phenotype in the SynGAP mutation. So this was done. This was given for only 5 days and measured on day 16 or day 17. So it is given only during the critical period of development. And for the anxiety, we were able to recapitulate what has been published for SynGAP by Gavin's lab as well as by Seth Grant's lab. Here you can see in SynGAP, they are less anxious, which is reflected in the number of times they enter the center. When we given the compound to the mutant, you can see that the compound has no effect on the anxiety level of the normal mice, whereas it has restored or corrected the anxiety to the normal conditions in the SynGAP mutation. So that was very promising when we gave it at the critical period. So this tells that the locomotion or motor function is corrected, as well as their anxiety level is corrected. And when we did the social isolation, that means whether they prefer an object versus a person, you can see that the wild type spends more time with the stranger one, whereas the SynGAP mutant is not really spending too much time between the empty jaw or the stranger one. It might be interpreted as, I really not interested in the object or in another mouse. I just want to be left alone. So that pretty much is observed in many of the patients with autism. And when we given the drug, it does not impair the functions in the normal mice, wild type mice. But the good thing is it is able to correct the phenotype. You can see that it is able to spend more time with the mouse rather than the object. So it is able to correct the social isolation deficits observed in the SynGAP mutation. When we do the social preference, that is with the older mouse versus a newer mouse, again, the normal mice is able to spend more time with the newer mouse, but the SynGAP mutant mice really didn't care. It's like, okay, I don't know which one is old or which one is new, and I really want to be pretty much left alone. And in the wild type, when you're given that compound, in the wild type, it has no effect. The wild type is able to perform as it's supposed to do. But sadly, in this particular experiment, in the social preference for this age group, we were not able to correct the phenotype. That is, when we're giving it only in that particular development time point, that is 14 days to 16 days after birth. Sorry, 10 days to 16 days after birth. So we were not able to correct the social preference. It still doesn't know which is the familiar one and which is the newer mice. And when we looked at the memory aspects, because learning and memory is one of the major issues in autism and intellectual disability. So normal object recognition, again, we tested it between the familiar object and the novel object. So the discrimination index tells you how much time it probably spent with the novel object. In the wild type, it spends more time with the novel object. In the mutant mice, it spends very less. In the SynGAP mutant mice, it spends very less time. When we gave the compound, the normal mice is still able to remember and discriminate between the older mice, and the older object and the familiar new object. Remarkably, the compound corrected the deficits observed in the SynGAP mutation. The SynGAP mice that received the drug was able to recognize and discriminate between the familiar object compared to the novel object. So that was really amazing. We were really excited with that. But we were, like I mentioned earlier, for Fragile X studies, when you give it in critical period, we expected some kind of positive results. But the challenging is what happens at the later time points. But that I will come to that in the next few slides. So the next strategy that we followed is, do we have to continue to give the drug in order to see the effect? Or the early strategy was, if you correct it only between this age group, is it sufficient to lead a normal life in the other stages? Or you have to start a little early and continue to administer the compound so that they can at least lead a normal life. For example, if you want to suppress epilepsy, they have to keep on taking the drugs every day so that the epilepsy level is reduced. So like that, we have to give this drug every day. And the duration that we chose is a kind of approximation in the human age, maybe starting from maybe 6 months or 1 year till 15 to 16 years of age in humans. And again, we did the same behavior experiments, and we looked at the long-term potentiation, which is a cellular correlate of memory, which I'm not going to show you for the sake of time. So again, when we did the open field test, so these experiments were done between 10 to 60, and they were measured so drug was given between 10-day to 60-day old mice, and the behavior was done when they were 70 days or 70 days of age. And here you can see the wild type mice, the normal mice is able to run only certain distance, whereas the SynGAP mutant is still showing hyperactivity. When we given the drugs to some of the SynGAP mutant mice, you can see that it was pretty much almost close to the wild type. It's not completely at the same level as a SynGAP mutant, but it hasn't completely corrected to the wild type level, but it's not as bad as a SynGAP mutant level. So it's come somewhere in between. This is what we call as a partial correction. And when we looked at the anxiety, again, in this group, the SynGAP mutant mice has shown less anxiety compared to the normal mice. When we given the compound to these mice, you can see again, there is a partial rescue, I would say, or correction happening in the SynGAP mutant, which is not bad. That means when somebody has, when you're standing by when you're trying to cross a road, for example, and we normally look at left and right to see if any vehicle is coming, and then we cross the road. But in this case, let's say in the SynGAP, in this mice, they don't really care, they just walk on the road. But when we give the compound, they're still somewhat able to understand, okay, they should stand for a few seconds and look for something and then walk. At least they should be able to sense that. That's what these two results suggest. And when we looked at the social isolation, the same result was observed like the previous age group for the normal and the mutant mice. When we given the drug, you can see that in the SynGAP mutant, it is able to correct the phenotype back to the wild type level. You can see in this SynGAP heterozygous mutant without the drug, they're not able to discriminate between an object and a mouse. Here, when we give the compound, the SynGAP mutant mice is able to recognize, okay, this is an object, this is a mouse. I want to spend more time with the mouse like a normal, healthy mouse. And when you look at the social preference, just to recap what we saw in the earlier age group when we gave the drug between 10 to 16 days only, here it is 10 to 60 days. When we gave the drug, it is able to correct the phenotype back to the wild type level. So that was really, really exciting for us, because we were worried whether the mice is not able to distinguish between one familiar Y mice versus a new mice, which is a major issue in the autism. And when we gave the compound here, you can see that the SynGAP heterozygous mutant mice is able to recognize between a familiar mouse compared to a new mouse, and it spends more time with a new mouse. And when we tested for the memory aspect, it was the same as the previous strategy. We were able to restore or correct the loss of memory back to the wild type level. So that was really exciting again, because this hasn't been done by many many drugs. We have only few drugs are available that can do this kind of restoration for this particular age group. But the next one is the most challenging for us. The one is, is the drug effective and can it be a really good potential therapeutic candidate when it can correct the phenotype if it was administered after the development of the brain. So in the hippocampus, the developmental time point is roughly here. Earlier, we were doing it between this time point, and we started from this time point and continued here. And one group is only within up to this time point. But the most challenging is what is happening after this time point. This is the time point when the hippocampus is considered to be completely developed. That is like for the visual cortex, pretty much develops by 2 to 3 years or even 5 years for humans. So when we administer the drug from 30-day-old mice to 60-day-old, that is pretty much like a month. Every day we were giving the drug, and that is the equivalent of the age group that we chose in humans. Again, we did behavior and LTP. The reason why we did LTP is we know in SynGAP mutation in the adult hippocampus, starting from this age group and little beyond these days, there is a deficit in LTP. There is no LTP formation in the SynGAP mutation. So we tested this again. I can tell you that it is correct to the wild type level, but again, I'm not going to show you the data for the sake of time. I'm just going to discuss the behavior data because that is more relevant and valid. When we look at the locomotor activity in SynGAP mutant in the adult stages, again, it was showing hyperactivity like the earlier ones. When we gave the drug, it didn't rescue, unfortunately. The locomotion wasn't rescued at all in the SynGAP mutation that received the drug. This was when the drug was given in the adult stages, pretty much like an adolescent stages, sorry, not adult stages, young adolescent stages, that is after the brain has developed. But when we gave this drug to the SynGAP mice and looked at the anxiety level, there was a partial correction. So you can see that it is not as bad as a SynGAP mutant, but it is somewhat different, and it is not as anxious as a SynGAP mutant, but it doesn't completely come back to the wild type level. And for social isolation, again, we were able to see some correction in the SynGAP mutant mice that received the compound. Here you can see that the SynGAP mutant mice that receive the drug is able to recognize between an object and a mouse, and it spend more time with the mouse. When we looked at the social preference, again, the SynGAP mutant mice that received the compound was able to recognize between a familiar mouse and a new mouse, and it was able to spend more time with a new mouse. So that was really encouraging. These results are really encouraging because at least some of the features are getting corrected when we give the drug. I would say maybe for a 10-year-old kid or even a 12-year-old or 15-year-old kid, they were able to at least correct many of the features or deficits that we normally absorb from the childhood days. And when we looked at the memory aspects, again in the novel object recognition test, this is really amazing. The SynGAP mice that received the drug, it was still able to remember like a wild type mice. It was able to distinguish between an older object and a newer object, and it was spending more time with a newer object compared to the SynGAP mice without the drug. Here you can see in the adult SynGAP mice that it was not able to distinguish between the old object and a new object. It didn't have that memory. Whereas the SynGAP mutant mice that received the drug was able to distinguish between the old object and the new object, and it was able to spend more time with a new object. So in summary, the compound that we have was able to correct the motor function and the anxiety when administered at the very early stages of development. Just for the sake of our understanding, I have put it as very early childhood. It's more of an approximation rather than correct age group. And here you can see in the next stage when we gave it from childhood days till the end of like 2 months of, you see affected, but the anxiety is in the novel object memory test. all 3 age groups was completely corrected the phenotype, which is really encouraging. And in the social discrimination, social interaction, again, all the strategies worked. That means it is corrected the phenotype. However, in the social preference, administration at the earlier stages did not correct it, but it has corrected the phenotype when it was extended beyond 16 days, and also when it was administered only in the young adolescent group. So that was really encouraging. And I didn't show the data for this. We have done the user level, and we have done it for the young adults, and that seems to be working well. And the data that I did not show is the synaptic functions apart from the chloride concentration that I discussed earlier. Here we looked at the LTP, which is the function correlate of memory, and we cannot really do at this age group LTP unless using a certain technique. So we really didn't test it, but we thought it's not required for this purpose. And when we injected from 10 days to 60 days and tested it in after 60 days, you can see that it is the LTP is restored to wild type level, and also in the SynGAP mutant mice that was given the drug from 30 days to 60 days of age. So these results are promising for us, because the most important table that we should focus, or the column we should focus, is this. After pretty much the brain development has happened, when we administer the drug, it is able to correct many of the deficits that we have observed in the SynGAP mutant mice. So how to take this compound forward? I know I've been talking to Mike, Pavel, and Sandra about the exciting research that we have. So right now, what are the things we can do? Basic research and find a therapeutic targets, maybe a potential candidate to correct the phenotypes, and then evaluate human iPSC lines, and that will be more close to the patient's sample compared to the mouse data, and validate most of the compounds, most compounds from the human iPSCs again in the mouse model, to see whether these two techniques is going to go back and forth. Some drugs, some of the compounds, if you have 5 good promising ones here, 2 might work and then go back again. Particular 2 steps will be kind of back and forth. And to enable things to move forward towards the clinical trials, we need to collaborate with researchers from other academic and also from the pharmaceutical companies, so that we can do these 2 steps really fast and evaluate it outside the current institute or even outside India, so that we can at least somehow aim to get some of these compounds in clinical trials as fast as we can. So currently with this compound, where are we? We are right now here. So we might need your help, suggestions on how to take it forward. And Mike, Pavel, Sandra, Daniel, they're all been in touch with me, and they're helping me to see how they can take this compound forward for further testing. So I'd like to finish by thanking my current and the past lab members. So this work was mainly work done by a graduate student, Vijaya, and Vijay did all the behavior analysis. He was blind to the genotype and also to the drug. We are confident that the analysis was correct. I think it's been roughly like 3 to 4 months trying to do the analysis. And Vijaya was very, very hardworking, very talented graduate student. She worked pretty much every Sunday for 6 months, I think, except 2 or 3 Sundays, like from 8 a.m. till 8 p.m. So she was very committed, very talented graduate student. And these are my collaborators. Ravi Manjudia, he's a very good friend and also collaborator. He's the one who has the drug. And Dr. Sridhar, he's from my institute. He's a chemistry scientist. So he synthesized the compound for us. And Ravi Manjudia, he's an expert on Fragile X syndrome. So we do collaborate to understand how SynGAP regulates Fragile X and how Fragile X regulates SynGAP. So with Professor Tapas, we're also evaluating some of the compounds that he has discovered that has never been tested on autism. So we are testing it. Hopefully, it also looks promising. Hopefully, we will have a convincing result maybe in a year or so. So thank you very much. And I'm happy to take questions.
Thank you, James. Hopefully you can hear me.
Yeah. Hi, Pavel.
Great. Hi, how are you?
Thank you.
Very much. Thank you very much. Just thank you for all of your work on SynGAP, frankly, over many years now. I think it's really making a big difference for us. And the results are really exciting, especially, I think a lot of the parents with older children, there's always a sort of uncertainty around whether any treatment would help their older children. And I think seeing something like this is so promising. So thanks for that. Okay, I'm just going to jump straight into the questions from the chat,
Sure.
and then we can go from there. So Dr. Kadam is asking, we know that males lag females in the developmentally regulated and age-dependent upregulation of KCC2 expression. Did you evaluate sex as a biological variable?
Thank you, Shilpa, for the question. So we didn't use female mice for behavior, but for the other experiments like LTP and the studying the chloride concentration inside the neurons. So we have used both the males, and I think we don't see any difference in the sex based on the gender. We do see the same level of changes in the male and the female, and also in the rescue level. Yeah. But okay. It will be interesting if you see a change, because people do say that in female mice there are some variations because of the hormonal changes that might disrupt the synaptic activity. But even when I sent my paper to Cell, we did the analysis between the male and the female mice. We couldn't detect any changes. It was just, they were pretty much matching. Yeah.
Okay, great. And then a question from Marta. She thanks you for the talk, and she says, do you think the compound has more effect in the hippocampus in all ages? Does it lose the effect to the cortex with time?
Yeah, so that's a good question. Like I said, we need to evaluate this drug even further. But based on the results that we got in hippocampus, and also from the earlier studies, my own studies in Gavin's lab, I wouldn't expect to see a drastic difference between hippocampus and the cortex. We should be seeing similar results. So that's my hypothesis, but we have to evaluate it. Yes.
Okay, and just a related point to that, the vehicle to administer the compound,
yeah.
and I guess passing the blood-brain barrier is something that Dr. Huganier mentioned is a challenge
yes.
with ASOs. What's the story with this compound in that regard?
So this compound was given intraperitoneally, but we have many derivatives, that's dozens of derivatives, which is water soluble, and after those we know few of them are even more effective than this particular compound. So that we need to test them by giving it orally, like mixing them in water or doing the gavage. To answer the question whether it crosses the blood-brain barrier, we have looked at it carefully by sending the brain samples to the companies. It reaches within 15 to 30 minutes, and it stays, I think, for at least a day or two in the brain before it gets removed. So it does cross the blood-brain barrier.
So it would be potentially something that would have to be taken on a daily basis, for example.
Yeah.
Okay, great. And then another big question: side effects. What sort of side effects have you seen?
Okay, so from my graph, it was like the normal wild-type mice. They are behaving normally, and some of these compounds are given for pretty much 50 days. If you look at the 10 to 60 day age group, and also for a month from the 30 to 60 day age group, we don't see any side effects. They eat well, they maintain their weight, because we do calculate the weight before we inject the drug. So we don't see any kind of side effects. Now, if there was any, we would have noticed that in the wild-type mice. And we did not see any death also of mice.
Okay, that's great. Very good. With regards to dosage, how was the dosage selected, and do you expect different results with different dosages?
Right. So sorry, just as a follow-up with the earlier question about the possible side effects, we haven't done the toxicology test like we do in the company. So we are looking for collaborations, because the companies with the good lab practice certificates, they can do the toxicology tests. So based on our, whatever we could do, we were sure that it doesn't have any side effects. And the administer the compound, we used DMSO. Like I said, it was not dissolvable in water, but we have made derivatives which are water soluble, and it is more effective. And the concentration was determined based on the target protein, how the compound was modulating the target protein. So based on that, we have taken the concentration. For humans, it might change a little bit, but again, after evaluating all the toxicology reports, then the experts will come and decide those things. Yeah.
Okay, great. Another question: has this compound been used in other diseases in humans?
Yeah, so I don't think this is an FDA-approved drug yet. So we are in touch with some companies who could take it forward, but right now, the answer is no.
Okay. And then a question from Camilla Morales: have you looked for changes in interneurons directly? For example, levels of PV or SOM?
No, we haven't done that yet. We would like to do it at some point. This is like a 1-year-old data. Still a lot needs to be studied. As of now, we don't know how this particular compound regulates the specific type of interneurons, like the paravalvular or some of the starting neurons. We don't know yet.
Okay. And one more question. Actual SYNGAP1 expression, is there any relation to that? You know, that's something we talk a lot about, again, with ASOs and other treatments. Is there any connection between that and the compound?
So if I understand the question clearly, you're asking what is happening to the SYNGAP levels when we administer the compound. So we have done those experiments. It doesn't modulate the SYNGAP level. So it is away from, it's not a direct contact of SYNGAP. Like I was listening to Richard Hugeney's and Gavin's talk. So they try to restore the SYNGAP level, but our approach is somewhat around it, because it's a genetic mutation. It's there from the embryonic stages. It's going to be like a uphill tasks to restore the SYNGAP level and bring the scientific function to the normal levels. So we were really focusing on anything apart from SYNGAP. So this compound does not modulate SYNGAP levels.
Okay, great. I think that's everything I see on the chat so far. I just want to say, as Dr. Clement said, we are working to try to get this going as fast as we can. We're trying to get some human cells over to Dr. Clement and also connecting to pharmas. So obviously we're very excited about this, and hopefully we can see more results in the coming months. Yeah, definitely, yes. Okay, thank you very much. Marta, I don't know if you want to sign us off.
I have a last question. Sorry, what you expect different to see in organoids?
So what's the difference we see in organoids in terms of the effect of the compound, or like I'm not really clear about your question. So you're asking, do we see the similar effect in the organoids when we administer the compound? Is that the question, or? Hello, Marta? Because there was so much background noise, I couldn't really catch it.
Marta, you're still on mute. Can you just clarify the?
So we don't know if the question is related to when we administer the compounds in the organoids. We don't know, but we would expect the same, similar results. Yes, that's my hypothesis, but we haven't tested it yet. So that's the reason we're looking for people who do work on human iPSCs so that we can take this forward.
Okay, great. Well, thank you very much again. This will be recorded and posted online for anyone who couldn't make it today. And we're very excited, and we hope to hear more from you.
Thank you very much for the opportunity.
All right, thank you.
Yeah, thank you. Take care. Bye.
Bye-bye.